이름 박소희. Okay, first off, probably you'd like to know what's the most important data, what's the average, right, of this thing. The average was 34.4 out of 45. Yeah, that was a class average. And the class median was 36. Class median means the 50th percentile. If here we have 100 students, then from top to 100, the score of the one who had the 50th percentile out of all 100 is what this class median defines. So usually, it's not always true, but usually the borderline between A and B is usually determined around at this class median. That's what you kind of expect. But one thing that you have to also remind is here we have a number of the international students, the exchange students. They are going to be excluded in your grade scale determination in the end. So here, all this class average and class median was added up together, including the exchange international student too. So your actual, the number could be different. But anyhow, I'm going to also put all these numbers in was it the portal? Yeah, Sookmyung portal, so that you can check. Also, the final exam score will be also available once the grading is done. And another thing, actually, the most important thing is, let me again apologize for this being late, the grading. This is exam week, actually. So hopefully, of course, obviously, I'm not going to be that late in the final exam grading, so you will be able to, in timely, you'll be able to get those scores for your grading and the scores available for you that I should. Okay, so another thing is, so this is exam week, and practically I'm just actually aiming that actual lecture I'd like to finish all those lecture material today, hopefully. So the Wednesday lecture, that what I'm going to do is, considering we are going to go over some of your questions, especially I will try to pick some of your student questions up until today, this scrap paper questions. Then I will select some of those questions actually strictly related with your exam content material, so that Wednesday, basically, I hope that we like to go over this session as a question and answer session, which is more related with your actual exam. The final exam that's what I'm trying to do, I'm planning on, so that I don't want you to actually waste too much of the time, especially this final exam way. So that's how I am planning on. Obviously, of course, that lecture will also be recorded. So if for some specific reason, if you cannot make it, of course, then you can try to review that on your own through this recorded version of the file. And another thing is about the makeup exam, because last Wednesday we had to cancel the class. I'm going to, because it's a policy, I'm going to provide some recorded lecture, but that lecture is not going to be part of your final exam. That will be just the extra. So later on, I'm going to that will be my old version of the lecture regarding the cell cycle and cancer. But that one, obviously, you will not be responsible for the final exam content. It's just for your own benefit. Okay, and if you had any question, specific question on the grading, why did I get this several points deducted on particular question? Obviously, of course, you can approach me and then we can discuss it. But in particular, the first question, the first essay question, that the way my rule on how I graded on the first question of the essay question was, because there are either way. Some of you say yes, some of you said no. Okay, whether the evolution is really essential for the goal of immortality. But mind you, that question, as I announced during the lecture, that question was directly from one of your scrap paper question after a couple of first lecture, that regarding the whole this purpose, whole this the life characteristic, common characteristic is because based upon that, for some reason, all these living organisms are like pursuing to the immortality. They want to live forever. Based upon that, the question was actually the student who gave that question, orienting. That question was oriented to that purpose as I translate that thing. But just because some of you said, no, it doesn't, it's not related at all with this, I didn't automatically deduct any point. No, I just followed the logic. Okay. So not all those students who said, no, it is not related, I just deduct the point. Some of you get the whole credit, even if you said, no, it is not related with any, this evolution is nothing to do with this, the immortality. Still, when I follow this logic, it made sense based upon those original questions, the philosophy, then I gave the full credit. And also, some of you who said automatically, yes, of course, it is related with blah, 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 although it was, those opinion agreed with my personal opinion, but just because it agreed with my own personal opinion in this classroom I did not give automatically any full credit. I also followed that, and if I just saw something, it is not properly, logically, it is not fit, then I deducted some 1 point up to. That's how I graded that particular question, because yeah, it was really subjective question asking what is your opinion, and then just because you automatically opposed to my opinion, personal view, I took 1 point off. No, it wasn't like that. So that's how I did try to make you understood. And also on Wednesday lecture, if you had any particular question about the content, although it may not really help in preparing for the final exam, because final exam you're not responsible for any material for with this. You can still ask a question freely too. My impression is, especially some of the essay question, I noticed that a lot of students simply seemed like followed some of the explanation version, answer version on the internet. So that's another like example that not always, just because some information was up on the internet or even ChatGPT said so, doesn't always is 100% correct one. Is okay. So, any question about the exam? So most of you actually did very well, very well. Our class high was, I think it was a 43, as I remember, 43 or 42. There was a class high. So obviously class low, I don't remember even. Okay. So today, we are going to finish off this whole biotechnology thing. So this is the PCR, polymerase chain reaction abbreviated with, is one of the most famous such biotech technique and one of the most widely used technique, important technique. And it was relatively recently invented. It was only in mid 1980s made available, and with over the years, throughout many refinement, of course, it became so much useful, important, and widespread technology. And bottom line is, what is PCR? As this name suggests, it involves the polymerase. It's a DNA polymerase. So this reaction is a DNA synthesis reaction. It's very similar to the DNA sequencing reaction. So somebody had an idea about, actually, there's a little bit of adaptation. DNA sequencing reaction is also, the Sanger DNA, dideoxy DNA sequence reaction is also trying to copy the DNA sequence of one such specific template area, is how they're trying to read the DNA sequence. As such, this PCR reaction is very similar in that certain target area of the DNA we designate as a template. At this point, I hope all of you at least understand the concept of a template. What is a template? Okay. Template is like example that you target out of which the DNA is copied or the RNA is being made, whatever. The template is such the definition. So template is something, the nucleic acid, we are talking about this, the template, nucleic acid template. Usually we refer this template DNA. So in transcription, remember? Transcription, the RNA synthesis being carried out along, trying to copy the certain region of the template DNA is transcription. This is RNA. In DNA replication, you are trying to copy the specific area designated as a template to double up, duplicate, or replicate. So that's how you utilize this template. And so in the DNA sequencing as well as this PCR, a specific template DNA is trying to be replicated. So to do so, you have to have this template DNA for the reaction. And this is the enzyme, DNA polymerase, for replicating DNA. And what is the dNTPs? The raw material, the nucleotide. So all these dNTPs will be incorporated along with this template DNA to copy this thing. And another thing is this. The important thing is, what is primer? So these are four, actually, the major important key ingredient in this reaction. And this is also applies the same ingredients are also needed in the DNA sequencing reaction too. All those DNA replicant reaction, these common ingredients are required. Okay. So template DNA you can understand, of course. This is something that we need to replicate or copy. Polymerase enzyme, obviously we have to have, because that enzyme is the one actually catalyzing this reaction. And dNTPs, obviously you have to have, because those are raw material, okay? The sources, resources that assemble along, according to this actual original sequence of DNA. But what is this primer? What is this primer? This is the description of, overview of the so-called PCR reaction. And here what you see is here's nucleotide, DNTPs, and some primer is illustrated over here. And can you, by looking at this diagram, can you understand the role of this primer? Primer is also very short piece of DNA. Primer is. Nucleotide is a single nucleotide. On the other hand, DNA primer is something like a 10 or 12 or 20 nucleotide, very short fragments of the DNA. But one condition, all those primer have one condition. What does it tell you? There are two different kinds of primer here in this reaction. In orange, both of them are in orange. Two pieces of, so to speak, primers are added in this PCR reaction. And this is a one round of the PCR reaction, so to speak. And can you understand the role of this primer in this reaction? Both of them seems like this arrow indicate what? This arrow, both of this arrow seems to indicate what? The direction of DNA synthesis. Now, just by looking at this figure, can you figure out the role of primers? Anybody but those the bioscience major who can answer? Obviously, of course, those who majors should know by now what the primers are. Primers are, so to speak, if I compare the primer, it's like a seed money. DNA synthesis cannot start without primers. So if you want to start any DNA synthesis reaction, like DNA polymerase reaction, in the event of DNA sequencing or PCR reaction, basically both of them are basically the identical thing. First hand, you need certain short fragments, short piece of this DNA called the primer. Without it, DNA polymerase cannot start. That's the characteristic, very distinct characteristic of DNA polymerase. All those DNA polymerase. Without primer, they cannot start working. So those primer, that's why I compare this primer as some kind of a seed money. Or if you don't like my version of the explanation, you can just think this primer as a stepping stone or something. Something that helps, that gets you started. So obviously all those primers should be complementary to the part of this primer. The reason why this primer stick to this part of this portion is this primer sequence is complementary to this portion of this template DNA. And the primer on top is also complementary to the sequence of, remember, in the beginning, the template DNA was provided in the form of double-stranded DNA. So in order to copy both strands here during this PCR reaction, you have to provide two primers going the opposite direction. Remember, all those DNA synthesis, also RNA synthesis, always proceed from 5 to 3' direction, always. So you have to provide the two different kinds of primer to facilitate this synthesis reaction. And another thing is you have to separate, because this original DNA template, the double-strand DNA, are held together, this double-strand DNA, through their complementary hydrogen bond. So you have to separate so that this primer can sneak into instead of their original pairing. Excuse me, and these primers have to get in the way of their original pairing. That's what this primer's role. And then once you have this primer, starting from the next of the primer, this DNA synthesis can go. And also this primer will allow the extension of this primer. So later, until they reach all the way down to the end of the primer sequence, then the reaction ends. That's how you perform this DNA replication. So in other words, you have to separate. It's called denaturation. You have to separate this double-strand DNA into single strand. And heat is very convenient for the DNA. By applying the heat, you can temporarily separate this double-strand DNA into individual single-strand DNA. That is a very convenient way of achieving this. And once you do, and then cool it down, we call this annealing. Cool it up, then during those cooling period, this primer will take the opportunity, then try to grab. Instead of original this pairing, primer will try to sneak into and then pair it. And then now, this primer can be used to drive the new DNA synthesis like that. That's how you go the DNA replication thing. And PCR is simply, so somebody named Kerry Mullins, this gentleman, had a kind of a crazy idea about, hey, how about I'm not interested in replicating entire chromosome or anything. That's not my goal. I just want to specifically, let's say this is, for example, an insulin gene in human chromosome. I am only interested in replicating this area, human insulin gene area only. Is it possible? How can I do that? Well, you can try to copy them by adding specific primer. If you add primer in this way, then the boundary of this primer, two primer set, will be replicated. Top and bottom strand will be replicated, right? By going through this particular step. So at the end of this one round of a replication reaction, if you do this, like one set of the two primer, obviously, if you provide this primer set and all these raw ingredients, then at least you will be able to duplicate this original single copy of this human insulin gene into two. That's the end result of this one PCR reaction. But obviously, as this name suggests, PCR reaction is not intended to simply try one single reaction. Then it shouldn't be called as a chain reaction. This chain reaction means it is going to repeat and repeat and repeat, something like 20 or 30 times of this reaction. Then just mathematically speaking, if you repeat this reaction, something like a 30 round of reaction, then how many copies? Starting from single copy of this gene, how many copies can you generate simply by repeating 30 times of this reaction? Huge. Huge, huge. You just calculate it. 2 by 2 by 2 by 2 by something, 30 times. Then you get the number. That's the power of this PCR reaction. However, one very technical obstacle is, so this explains the nature of this primer. Forward, and usually called the forward and reverse primer. So whatever, you can call this as a forward and this as a reverse primer, whatever. But simply suffice to say that there are two opposite directions of the primers are needed for this PCR reaction. Is if you understood, there will be just enough. Specific characteristic of a primer, they are complementary to the sequence of the blah, 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 like that. Okay? Unless you are actually practicing the PCR reaction in the laboratory, you don't really have to worry about this. I have witnessed so many students make mistake in getting the PCR primer sequences, wrong PCR primers, but it's okay because you are not the one who actually trying to practice this thing. But anyway, this is a theoretical background of this primer nature. So like, for example, if these two sequences are the template sequence, right? Then you can try to practice. You can try to write down two primer sequences to duplicate the entire region, copying this entire region. You try to practice at at home or anything, then you will get a little bit get the hang of it. So anyway, if you do so, only by repeating 3 times of this PCR reaction, you get the 8 copies. So if you repeat this 30 times, then you will have a huge number of these PCR copy numbers. Okay? Millions and millions of copies can be generated. However, you have some technical problem. This is what it takes to go through such a simple one round of this PCR reaction, because in the beginning you have to do this. Separate. At first, you have to separate the template DNA by applying heat, usually 95 degrees Celsius. Then template DNA will become single-stranded. Then now you lower the temperature into reasonable, something like 55 degrees Celsius or 65 degrees Celsius or whatever. Okay. Then at this point, the primers will get into each specific location of the primer, and then now they are ready for DNA synthesis. And DNA polymerase will allow this extension. Some of you may be a little bit of a surprise why this extension is the period which this DNA replication reaction is going on. But then surprise, 72 degrees Celsius? This is too high, usually. This is not usual temperature for any enzyme, which is we accustomed to know that about 37 degrees Celsius is probably the optimum temperature. 72 degrees? Wow, you can't. No one can. No enzyme can survive this high temperature, 72 degrees. If you if you touch the 72 degree water bath, probably, it's too hot. You cannot. Why this PCR diagram have this type of a high temperature? It's because one technical problem is this. Each different round of reaction, this DNA polymerase reaction, you have to apply this high temperature and cool down. But once you apply this high temperature, then all those enzymes that you have provided will be killed. DNA polymerase cannot survive. DNA polymerase is a protein. Protein is very, very sensitive to high temperature. So all those structures will be destroyed. So initially, actually, what they have done, the scientists, one round, the first round is over, then probably all those DNA polymerase enzyme must have been killed. So then you add another fresh enzyme in the second round, and then go through. And then at the end of the second round of the reaction, all those added DNA polymerase must have been destroyed. So you add another wave of polymerase. Originally, that's what they did. But imagine, you cannot really go this thing like that. It will take forever, and who can just stay all night and then do this tedious thing? That is not going to work unless you come up some very revolutionary thing. Was this thermostable DNA polymerase enzyme? If we could, I wish we had some DNA polymerase which can stand something like 95 degrees Celsius. Then we don't have to worry about killing at this round. We can just go all the way, repeating over and over without, in the meantime, intermittently, we add another fresh round of this enzyme. We forget about that. If we somehow can discover some imaginary magical enzyme which can stand such high temperature like 95 degrees Celsius, that will be really, really great. And then he did. That's exactly what he did. Like in bacteria, who else? Certain weird bacteria like archaebacteria lives in hot spring or volcanic area, like 100 degrees Celsius. They don't die. That's their optimal habitat environment. So then they found out some of the DNA polymerase enzyme, which has actually the optimal temperature at about something 70 or 80 degrees Celsius, and it doesn't get destroyed even at as high 100 and beyond the high temperature like that. So that's we define such enzyme as a thermostable DNA polymerase. There are a lot of those different kind of thermostable DNA polymerase. But anyway, the most famous and the first ever found thermostable DNA polymerase was a PEG. It was found from one type of archaebacteria which live in such a high temperature environment, as I mentioned. So we store such magical DNA polymerase and then get this PCR reaction so handy. And afterwards, this PCR reaction became so famous, so convenient, so widespread, can be applied to almost every the molecular and other medical technology. So even thanks to this coronavirus pandemics, a lot of us even heard this so-called RT-PCR. The only difference between RT-PCR and regular PCR is here in regular PCR, you are trying to replicate the DNA template. But in RT-PCR, your target is RNA. You're trying to replicate the RNA. How? There is another technical issue in trying to tackle the RNA. RNA is very unstable. So to do so, you have to first convert this RNA into DNA. How? Remember some of the viral enzyme that convert this RNA into DNA. It's called the reverse transcriptase. Certain viruses have it. and utilizing that enzyme too. And first, you convert this RNA into DNA and then perform the regular PCR. That's exactly RT-PCR. But that type of technique like RT-PCR is very useful in like trying to detect some of the RNA copy, like just like why we used RT-PCR in the coronavirus cases. Because coronaviruses are RNA virus. So if you are interested in, did we have the traces of coronavirus in our body or not? And because this technique, like what's the power of this PCR? One word, can you characterize the power of the PCR technology just by one vocabulary, one single word? Anybody? PCR. Why PCR is so powerful? Proliferation, similar, kind of close. Amplification, like someone spilled their blood stain on the crime scene. Just blood stain, one drop of blood stain cannot provide any information at all about. But out of, by scraping this one blood stain, I mean, can you imagine that what kind of DNA? The traces of DNA probably must have there. But by applying this PCR technology, you can hugely amplify the amount of DNA and then easily identify. Simply you can sequence and all kind of other characteristics of this DNA. Then you can identify, oh, this blood belongs to somebody. Saliva too. And for example, that's how you were able to also complete the Neanderthal sequencing technology. You found some tiny bone, the fossil bone of Neanderthals. How can you get any information of this, their entire genome? You collect a bit by of such very trace of the information and amplify all those DNA and then reassemble, analyze, and then complete. So that's why the PCR technology revolutionized all those different technology relating this life cycle science and medicine and diagnostics and forensic sciences, and you name it. That's why no wonder No wonder actually this gentleman, initially, after invention of the PCR technology, this was original machine that he sold. He quickly found his own company and then sold the special machine, was making money. Even it seemed like he was not even interested in any science. He was seems to be more interested in making money on a businessman. And this type of original machine cost something like more than $30,000 or $40,000 at the time. Huge expensive machine, and everybody, all the labs in the whole world wanted to have one of them. So no wonder he became quickly a billionaire, made a lot of money. And people said, okay, you're enough. Because as a scientist, for whatever, you made a lot of accomplishment, quite a good accomplishment. However, your accomplishment was already rewarded in the form of this huge monetary reward. So no one ever thought about, don't even think about it and getting any honorary things like a Nobel Prize. Okay. You just made money. Don't think about this getting Nobel Prize. Nobel Prize is not for this type of dirty, worldly achievement. But about 10 years after, he even won the Nobel Prize too. Because the impact was too great. The impact was too great to ignore his accomplishments too. So even though he was already huge, a billionaire, but there's no way but to simply recognize his achievement in the form of this Nobel Prize too. That much impact in every bit of this scientific area, the PCRs, the effects was huge, was great. Okay, so and although there are several other very important technologies, but obviously we cannot go over every bit of things. So some of them I intentionally, I wish I could cover, but I had to include some of the cellular technology like stem cell technology, especially induced pluripotent stem cells like a new and promising technology. But I wish I could cover, but there's no time for us to go over. So finally, I'd like to go over one last technology throughout this another perspective of this viewpoint regarding this technical advancement. Here is very familiar figure, the procedure for imaginary and usual, like the evolutionary step for modern human, starting from the like chimpanzee like hominid. But what about this? What is this? Some people portray this may be our future in the course of evolution. So this is always a like very big discussion point for us. Aging, no one wants to get old and no one wants to die. So always, if someone trying to have some good news about this aging and death, and usually, yeah, it just draws huge and sharp attention of everybody, right? Because of this, these are the two. No matter how much money you have, and no matter what kind of achievement socially or like in business you have, but these two things you cannot escape from. And look, it's very silly thing. Time magazine, the cover page of some old thing. In the year by 2045, you will be free from this. Getting old and having to die, you don't have to. So you will be able to live forever by in the 2045. I don't buy it, of course. In the the year 2045 is like it will be coming too soon. We have only about 20 years left, and I don't think in 20 years all this problem of aging and that death can be solved. So this is a really too rosy outlook. But anyway, still they have some point. Their point is this always led this economic growth, and those economic growth resulted in a lot of these resources abundance. Okay. And thanks to that, then usually it follows this the quality of our lives has been improved. That has been always the case. So, however, if you look back, this our modern human, like it's been estimated that about 200,000 years ago, like this similar form to the present form of the modern human appeared. So our history, in other words, our history is only about 200,000 years old. Okay. Then what about our life expectancy? This is the red line indicates the change in our human life expectancy plotted against the years. So, like, even until like 15th century or anything, the human life expectancy was only about 20s. Yeah, obviously. When it says that your life expectancy is only 20 years old, that doesn't mean that by reaching the 20 years old, you will be expected to die. No, it's not. The reason why it has such a low life expectancy is there are so many early infant moral rate and those people who die so early ages due to illness, starvation, and the war. There are so many people die in very early the stage of their life. That's why it has such a short life expectancy. Some, even in these days, some people were lucky enough to live something by up to their 70s or 80s, of course. However, there are so many people who die early simply had made this type of, on average, this is such a short life expectancy. But then it skyrocketed only very recently in the mid 20th century. And to help understand why this trend, because this red line is life expectancy, oh, sorry, my bad. And this blue line indicate the economic growth in terms of GDP per capita, right? Economic growth. So in other words, as economic growth expand, then so does this life expectancy. So in other words, this life expectancy is closely related with this economic growth. It is approved. So we will only, thanks to this huge economic boom, after this industrial revolution and etc., technological advances that followed, only very recently we were able to enjoy this kind of a very stabilized and also long life expectancy. That's the fact. And especially now, there are two very, very, very promising technology emerged very recently that can even further enhance this trend of human longevity, which are, one is this human genome sequencing that we covered in the last lecture. Why? Why this genome sequencing has anything to do with increasing our life expectancy? Strange. And genome editing is the one that is the new technology that I am going to cover today. So first, let's take a look at this genome sequencing. The technical aspect we have already looked over in the lecture. Let's take a look at this cost side of this human genome sequencing. In the beginning, it was very, very inefficient and very expensive. No single individual could even dream of having their DNA sequence all discovered, figured out. Okay? But even as you can see, in the year 2000, even there was the first ever individual DNA sequencing was even succeeded, the James Watson's genome. Why? Because the cost of DNA sequencing initially, even in the year 2002, if you want to actually have the DNA sequencing, your entire chromosome DNA sequencing, then it would cost $300 million. Oh, that is a very good fortune. Okay? So then it is not affordable. However, drop like this. So the projection by the year 2020 became even under $100. It was a little bit of like also a little bit of exaggeration. Still, we are not here yet. Even in the year 2024, the cost of the individual human genome sequencing it's not really under $100. Under $100 is like even below 10만 won. Oh, then we can even, but it's not really that low, but still it's very close. So in other words, the projection is in 5 or 10 years, within 5 or 10 years, this cost of sequencing entire individual DNA sequence, the DNA sequence will be even less than $50, I guess, usually. Then everybody can do that easily, casually, get their individual DNA sequence read. And what is it so significant? Now we can change the entire the approach to address any illness or disease. So such affordable DNA sequencing, technically and also cost-wise, allow this new approach of so-called genome-based customized medicine. Conventionally, up until now, we had only available, the cure was provided in the form of this standardized and reactive. These two are keywords to the currently available way of curing the medicine. Standardized. Standardized means, yeah, we have already some specific medicine and the method to cure any specific disease or anything. But if you, as an individual, if you had your own specific some illness, but luckily if your illness falls into already standardized way of curing, then you are lucky, good. But for some reason, your illness doesn't belong to any of those categorized, standardized way of curing, then, oh, sorry. There's no way we can take care of you. So that's a standardized way of medicine. And reactive is, you can only, once you discover, only after discovering that you had something wrong, then you go to the doctor and, hey, I have this uncomfortable things. I may have some cancer or anything. Then you should try to find out what's wrong and is there any cure. That's we can label as a reactive medicine. So this standardized and reactive is the conventional way of curing the illness. And we can change this thing into now not standardized but customized, personalized. And also, instead of reactive, but we can change those approach into preventive or preemptive way of. So you haven't developed any illness yet, but by just examining, by sequencing your DNA, it's like, oh, there is a very high chance that you are going to develop diabetes in next 5 years. So you better do something about it. Type of thing is possible now. Already, some of the celebrities have this, and then they just took the preemptive measure to try to prevent some of the specific type of cancer and overseas, as you remember. So that's such an example of this preventive medicine. So those DNA sequencing, which is so like prevalent and then popular, widespread, so that every individual can easily, affordably do this DNA sequencing, just like you go to the doctor's office and then do casual routine test, will make this type of different way of handling any illnesses. But just because you found some probability of developing some specific illness doesn't really mean that you have some specific cure, very specific cure for that. No. So DNA sequencing itself alone can only provide some early and thorough detection But DNA sequencing alone cannot provide any actual cure for this. But luckily, this new technology called genome editing technology emerged very recently, in the last 10 years, right? Can actually provide this. It's called CRISPR system. Some people call this as a CRISPR genetic scissors because just like scissor or a pincet, you can take very specific point of DNA on your chromosome and then you can manipulate, which means you can change those DNA into something that you desire. It is a new technology. So here at this point, this CRISPR technology, what's the difference between this CRISPR genome editing? It's called the genome editing technology. What's the difference between this and that famous GMO or LMO, GMO technology? GMO, those conventional GMO technologies trying to insert foreign gene into any random place of your genome and taking advantage of the power of that foreign gene. It can have, depending on what type of gene is, the foreign gene inserted, it can provide so many different effects. But still, that gene itself was imported from outside source. Okay? So you cannot really have a very specific control over. That's the drawback, huge drawback of this GMO technology. However, this genome editing technology, your own gene can be corrected. It's not something that you are trying to insert any foreign gene, but your actual DNA sequence can be corrected. It's the actual new power of this technology. So, anyway, this CRISPR, why this name sounds so weird, the CRISPR? It stands for actually this long, huge name. Clustered regulatory intensive short, palindromic repeat, whatever. Okay, that's the abbreviated form of this. That CRISPR stands for this abbreviated form of this entire full sentence. To simply put it, it is an acquired immune system of bacteria. Bacteria's defense system, as we have already once looked at the other type of bacterial defense system, which was what? Restriction enzyme. Restriction enzyme was bacterial defense system against the invading virus, wasn't it? The scissor-like restriction enzyme. But bacteria also have additional, very specific, just like our antibody. The mammals have a specific defense system called antibodies. Antibodies' power is very specifically, antibodies can recognize, the antibodies can remember the foreign invader once invaded in the past and then elevate and speed up the response, defense reaction so rapidly, and that's the power of these antibodies. And so does this CRISPR system in bacteria. They remember. This CRISPR system is also against the virus, but unlike this restriction enzyme, the defense system of bacteria, this CRISPR system remembers the specific virus sequences. So, if the specific virus invade again, and this CRISPR system is activated and so efficiently get rid of them, just like our antibody system does. So anyway, this is some figures illustrating the mechanism of this CRISPR system. I don't really have to go over too much details about, but anyway, a little piece of the viral DNA, once every time the virus invade, then the bacterial host collect some piece of virus DNA and keep them in their genome. And then later, if the same virus invade again, then this some part of specific part of DNA is being activated and then being used to inhibit this viral DNA, which I don't have to really go over very specific technical detail. That's how they do. They just cut very specific DNA sequence of the target. So this CRISPR system is more versatile and more specific than the conventional restriction enzyme system because any very specific target sequence itself specifically recognized and cut by this CRISPR system. So the utility of whatever, okay, good for them, good for bacteria, because they have this kind of a defense system. Very good. However, we don't care. Who cares? Well, we only care our own interest, human, very selfish human. So the great utility of this CRISPR system for us is we can utilize this as just like a very convenient tool allowing this find and replace. Okay, in our genome, I have some very specific DNA sequence, but I don't like this DNA sequence. Okay, then can we just replace this just like editing? That's why it got its name or genome editing. Some of the specific DNA sequence, you found that, oh, this DNA sequence has mutation. So because of this mutation, you have a very likely chance, very high chance of getting cancer because of this specific mutation in your DNA. So is there any way we can do about? Oh, sure, maybe we can try to change this sequence into something like the wild type or normal DNA sequence. It allows, this CRISPR system allows actually. Although there are, to do so at the stage of adult already, developed adult, you actually, as you can imagine, there are several actual technical obstacles that you have to overcome. However, at least it's there. You can do that. And actually using this type of technology, although there was one case that at the early stage of embryonic development using this CRISPR technology, and scientists in China attempted to correct or edit the baby's gene into something else, and then it showed some success. But then the scientist was expelled, totally banned from practicing any science afterwards. But anyway, technically it's there, you can do it. And actually for all type of other animals and plant crops, this CRISPR technology have been already heavily applied, and we have so many different diversity of such already edited different plants and animal species we have created already. So anyway, not only that, you can also try to, if you are interested in some function of any specific gene, you can deliberately mutate that particular gene using this technology. So bottom line is, this CRISPR technology can be used in creating mutation specifically, and also correcting the DNA sequence into your desired. These two directions are the two main directions that you can use this CRISPR technology. So, technical, once again, it's technical thing. Probably here and there in some of the internet, you may have seen very similar this figure illustrating this CRISPR. Anyway, target gene can be specifically cut. That's the only point. Target sequence can be specifically cut. And once it's cut, you can automatically generate a mutation. And also, if you don't like, you can even try to replace this area into your own version of the provided one. That's how you can uh, achieve both mutagenesis as well as gene replacement too. Obviously, of course, then no wonder they won the Nobel Prize for this technology, and relatively very recently, like that. These two ladies were the one to get the most credit for this technology, although there are several other scientists also who have contributed so much about this. But anyway, so we can utilize this. So these two technologies, if these two technologies, powerful techniques like genome sequencing and CRISPR genome editing can be combined, imagine what kind of thing you can try to achieve now. You can detect some of the unusual things on your genome, and you can change those into. So it all applies to those any traits that you have discovered, for example. Imagine, let's say you have discovered a gene, a trait, a gene that is responsible for the trait of your intelligence. So you have somehow figured out that if you have some specific DNA sequence in this trait, in this gene, making this your intelligence, then you can achieve very high intelligence like IQ over 250 or something, genius like level. Then you can try. You can try to change. Or imagine you have discovered finally a specific gene that making your height something like 6 feet automatically. Then you figure it out. You can easily identify what kind of DNA sequence is responsible for such desirable trait. Then you can mimic. If you happen to be the one not carrying that particular sequence, you can try to, you would want to change your DNA sequence into this. You can change your phenotype. Now changing your phenotype was something that the natural selection was the one. But now with this technology, you yourself can change your phenotype. And actually you can derive your own direction of evolution. Isn't it scary? So now human actually, technically, on the verge of acquiring this actual, the power of manipulating your gene and change it into this thing, into something that you desired. Now, can you predict the outcome? In which direction can we go using this type of technology? Like, so what about in terms of, for example, the genetic diversity? Like everyone, if different individual have certain kind of a standard, desired trait, then our whole genetic diversity eventually will all be lost. Everybody will look very similar, very good looking. Everybody's height will be very tall. Everybody's intelligence will be very, very like a super intelligent type. Everybody's physical ability will be something like that of the professional athlete level. Would be this type of trend. Are we moving? Or even with that, still, like every different individual's preference is so different, so we still will be able to maintain so much genetic diversity? It's probably a very interesting question, mind-boggling question. Here we are at, in terms of technology, here we are at, and probably at this pace, probably the technical advance will be so much more rapid and beyond our imagination within short distance of the future. Then are we ready for such kind of a different turmoil is my final word of this the class, should I say, actually. So, good. I was able to finish this thing. So on Wednesday actually lecture, I don't have to have I don't have to maintain this any scrap attendance scrap paper anymore because it's all like you're welcome to the class based on, and if you can make it, or so be it. You don't really have to come to the class because it is, once again, like I said, it is a final exam week. But if you come, then I'm going to go over some of the questions, as I said, is what I'm going to do. So long.